This is a simplified animation of going from the DNA level, which is a gene, all the way through to a protein. Let's start at the DNA level as represented here. The DNA is made up of a gene, and a gene consists of both exons and introns. The regions that are in blue are those that will be transcribed. The regions that are in red will be both transcribed and translated. So let's have a look at how that happens. First of all, we need to make a transcript of this gene. And it starts at the start of exon 1 at the transcription start site and ends at the end of the last exon at the transcription end site. Once we have made our first transcript, this would be called the pre-mRNA, as it is an RNA copy of the complete gene, exons and introns. What we then need to do is process that pre-mRNA to make our mature mRNA, and in doing that, we actually splice out the intron regions. So the processing involves removing all the RNA sections that were originally coded for by introns. Often the RNA has a poly-A tail attached to it. This is thought to help transition the RNA from inside the nucleus to out into the cytoplasm. Now we have our mature mRNA. A region of this will code for our final protein sequence, and that is the region I've drawn in red. This region in red here will be translated, so it contains the codons, each codon coding for a single amino acid. The process to do this is called translation, and as you look, translation begins at the translation start site at the start of the red region and ends at the stop codon, which would be at the translation end site. A single amino acid is coded for by 3 bases within the RNA, and it moves along until it reaches that stop site, and then you get the final protein. So, summarizing from the top, you go from the DNA level, which contains your gene, to a pre-mRNA, which is a complete RNA copy of your gene. You then process that RNA by splicing out the introns and adding a poly-A tail in most cases to get your mature mRNA. And this is what is used for a template to code for your protein, which is made of amino acids. You then translate that mRNA into protein and get your final protein product.